So our lab is studying cancer biology, and specifically we're trying to understand how the complexity of tumors affect the way patients respond to anti-cancer therapies. Tumors are very complex. They have not only cancer cells, but many other things like immune cells and blood vessels and fibroblasts and nerves. And our lab is looking into another very interesting component, which is bacteria and fungi that we and others found to be present inside tumors. So our research tried to characterize these bacteria mostly and fungi in these tumors, but mostly to understand what are the functions of these bacteria, how do they affect tumor biology, how do they affect the way patients respond to therapy. And we hope that by studying this, we can come up with novel ways to treat patients, because most of the more we study it, the more we understand that these microbiome, or bacteria and fungi as we call them, they really affect the way patients respond to therapy. And if we can modulate it, we can make patients respond better to therapy. I would say that the most exciting is the fact that for many, many years, even though cancer has been studying for many, many years, we didn't really recognize the fact that there are bacteria and fungi and viruses inside cancer. So I would say that this angle of cancer biology have not been thoroughly studied. But in the last few years, it's becoming more and more clear that these bacteria and fungi in the tumor has really can really dramatically affect cancer biology. So it seems that there's a lot to uncover. There's a lot of new things to understand, lots of low-hanging fruits, that by studying this for almost the very first time, we can really uncover a truly exciting piece of new biology and find completely novel ways to affect patients' response to therapy. And for me, this is very exciting. I think that the more we learn, there's more opportunities we have in modulating the response to therapy. One way to think about it is we can, kill bacteria, let's say, by adding antibiotics to other treatment options. But there's another way to think about it, and this is maybe we can not kill bacteria, but actually modulate the activity of these bacteria. So one can think of targeted therapies that are not directed to human proteins, as usually have been done, but directed to bacterial proteins. So one can think of a way to modulate specific activities of the bacteria, which in turn will modulate cancer, biology, and then in turn affect the way patients respond to therapy. So there's almost in my vision, there's new targets here that are just now being revealed, that the community can now start targeting and having completely novel ways of treating cancer. So the way I became interested in cancer bacteria was a complete serendipity. I was looking at the way that stromal cells, which is like normal cells that are present inside cancer, the way that they affect cancer cells' response to therapy. And I was co-culturing, meaning putting together cancer cells and normal cells and testing how the normal cells affect the way cancer cells respond to therapy. And I found this one unique case in which cells that coming from the skin of a woman drastically affected the way pancreatic cancer cells respond to chemotherapy. And I was looking for the mechanism, and after a lot of research, we found out that the cells were infected with bacteria. It was actually the bacteria that changed the response of the cancer cells to therapy rather than the stromal cells, the fibroblasts. And that made us think, me and the other people that worked with me, one, how can bacteria change the fate of cancer cells, the response to therapy? And two, are there really bacteria in pancreatic cancer? And if yes, that can be really important. So that's made us look into pancreatic cancer, discover that there are bacteria in pancreatic cancer, and so on and so forth. So it's really a completely serendipity. So, receiving any grant is really great, and receiving ICRF grant was really great news for us, because without going into much details, we have suggested I would say, a very high risk, high gain project. And it's always more difficult to raise money for these high risk projects. But we truly believe in this project, and we think that we can find something really exciting there. So getting the funds from the ICRF for this specific project give us the opportunity to really pursue our curiosity and our hypothesis, and hopefully in a few years from now, we can make some significant discoveries based on this ICRF funding. So for us, it's really been good news.